Good morning children. Today we are going to study about fermentation. In the previous lecture we have discussed about glycolysis, in which we have studied that the glucose molecule is converted into pyruvic acid. Now one glucose molecule gets converted to 2 pyruvic acid. Now let us study about fermentation today. In fermentation, it is actually the incomplete oxidation of carbohydrate, or you can say glucose, which occurs during anaerobic conditions. Now by set of reactions where the pyruvic acid is actually converted into ethanol, and this happens in case of yeast cell. In certain other cases, as we'll see later on, this pyruvic acid gets converted into lactic acid. So now let us discuss them. Now as you can see, in case of yeast, what happens? This pyruvate, you see pyruvate is a 3-carbon compound. Under the influence of an enzyme called pyruvate carboxylase, where the carbon dioxide is eliminated outside, it changes into acetaldehyde, which is again a 2-carbon compound. Now this 2-carbon molecule, along with the another enzyme, when it reacts, that is called as alcohol dehydrogenase, it gets converted into ethanol, which is again a 2-carbon compound. You can see ethanol as such. Now here the pyruvate is getting converted into ethanol in the presence of 2 enzymes which takes place. That is, 2 enzymes reacts. One is called as pyruvate carboxylase and another one is called as alcohol dehydrogenase. Now yeast poison themselves to death when the concentration of alcohol increases to about 13%. In both lactic acid and alcohol fermentation, not much amount of energy is released, and very small amount of energy is converted into ATP. Less than 7% of the energy in glucose is released. Now here we compare both the formation of ethanol as well as lactic acid. As you can see, here you see, in case of ethanol, glucose, while getting converted into pyruvic acid and later on to acetaldehyde, an enzyme is used which is called as pyruvate carboxylase, as we have seen earlier also. Then again acetaldehyde is converted into ethanol under the presence of an enzyme called alcohol dehydrogenase. Here you see the difference. During the process, pyruvate carboxylase, carbon dioxide is getting released, and again here NADH is getting converted to NAD+. In case of lactic acid formation, glucose, after getting converted to pyruvic acid, an enzyme, only one enzyme is used up here, that is called as lactate dehydrogenase, which converts NADH into NAD+, and the formation of lactic acid has taken place. See, both of these 2 reactions, they are produced, both of these 2 products, ethanol and lactic acid, they are produced from the pyruvic acid as such. But since the kind of enzymes are different, and that is the reason why the product becomes different. Okay, now let us talk about the homework assignment. Define fermentation. So fermentation, we have already discussed. Fermentation is a process in which the oxygen is not used up, and in this process, in absence of oxygen, the carbohydrate gets converted into either alcohol or lactic acid, and very less amount of energy is produced. So differentiate between fermentation in muscles and yeast cells. That I have already told you, the product is different since the enzymes in both of these cases are different. How fermentation is different from aerobic respiration? See, aerobic respiration will be covering later on. In case of aerobic respiration, this pyruvic acid The complete oxidation of these molecules takes place in the mitochondria, releasing much more amount of energy. Various enzymes are used up in Krebs cycle, which takes place in mitochondria, and hence the amount of energy released, or you can say it is complete oxidation, and hence the amount of energy released is higher. So that we'll be discussing later on. Thank you. Have a nice day.